Thank you for your interest in our paper on evolving neuronal plasticity rules using Cartesian genetic programming. In our group, we are studying learning in the human brain. It is widely believed that learning in biological agents happens by changes in the coupling strength between neurons, which is also termed synaptic plasticity. Until now, models of synaptic plasticity are mainly built in a manual process guided by researchers' intuition. Our goal is to find good plasticity models by automating these procedures, setting the update function of synaptic coupling strength, or also called synaptic weight, as an optimization problem. In other works, such a function is searched via parameterized functions or ANNs as so-called plastic neural networks. We, in contrast, are interested in finding free-form and interpretable rules by leveraging Cartesian genetic programming. Here, we study a simple all-to-one neuronal network with a linear activation function. In this unsupervised learning setting, the set of plasticity variables just consists of pre- and postsynaptic activity and the current synaptic weight. In this problem, we are interested in finding learning rules which align the weight vector with the first principal component of the data. Therefore, we define our fitness function as the angle between the weight vector and the first principal component minus some regularization term. The overall fitness is then averaged over different datasets. We have three different types of runs with datasets sampled either from T0, thus having arbitrary covariance matrices, from T1 with strongly correlated inputs, or from T2 with weak correlation and one dominant axis in the principal component. As a baseline, we used the manually derived Oya rule. For datasets sampled from T0, 5 out of 6 runs evolved to learning rules that perform comparable to Oya's rule, as you can see in figure A. 2 of these, learning rule 1 and 2, contain Oya's rule, as well as a learning rate scaling and additional factors in the case of learning rule 1, where these additional factors simply lead to noise in the weight updates, but average out over samples. In panel B, you see how these final rules performed, evaluated on different datasets from task family 0. Rules discovered with data from T1 or T2 specialize and outperform their more general counterparts, as you can see for learning rule 3 in panel C and learning rule 4 in panel D. From the rules we are looking at here, learning rule 3 is the only one that does not contain Oya's rule as a sub-expression. Having it as a closed form expression allows us to study why it still performs well on datasets with strongly correlated inputs. We calculate the expected weight updates, and we can see it in the formula. And in the case of a two-dimensional system with 2 inputs, the system has one stable fixed point, which is close to -1, -1, as you can see in figure E. The basin of attraction covers the whole initialization space, and thus the fixed point is well aligned with data sampled from task family 1. However, it is also the case that the fixed point remains close to the diagonal for data from other task family, where it is not necessarily well aligned with the first principal component. We can say that our evolving to learn framework can successfully discover interpretable biophysically plausible plasticity rules, such as variations of Oya's rule, but also rules that are specific to the used task family that exploit statistical properties of the input data. Whilst thinking in machine learning terms, this could be interpreted as overfitting to data, we actually believe this is a strength of our approach. We hypothesize that inductive biases to specific input distributions encoded in plasticity mechanism can be a basis of fast learning. As an example, humans, we are very good at learning fast from natural images, concepts such as what is a dog? But on the other hand, we're very slow or even unable to learn from other distributions, For example, distinguishing different QR codes. In this work here, we worked at purely unsupervised learning. However, in the brain, there are many more signals influencing plasticity during learning. It is difficult to build models on how these signals shape learning, how they interact, but we can now leverage our system by introducing these additional variables and let the evolutionary algorithm figure out how to make use of them. Thank you very much.